In this video, we will be discussing a portion of the root called root hairs and how they are formed. We will discuss this process by answering 3 questions using the Arabidopsis plant as our model organism. When we think of roots, we think of the underrated part of a plant that does all the work behind the scenes. We don't always see them, but they play a major role in plant survival. Roots are the part of the plant that attach it to the ground, providing it with structural support, nutrients, and water. Root hairs are tip-growing, tubular-shaped outgrowths that help anchor roots, interact with soil microorganisms, and assist in the uptake of water and nutrients. They can also interact with mycorrhizae, nitrogen-fixing organisms, and diverse pests and pathogens. Their formation occurs as a series of developmental processes, starting with cell fate specification in the root apical meristem and ending with root hair tip growth. Question number 1. What is the developmental process of forming root hairs? The radial pattern of a mature Arabidopsis plant has concentric layers of epidermis, cortex, endodermis, and pericycle surrounding the central vascular cylinder. The epidermis is divided into trichoblasts and atrichoblasts. Trichoblasts are able to form root hairs and are positioned over the junction of 2 underlying cortical cells. The proteins involved in root hair formation include H cells and N cells. In the epidermis of the root hair cell determination is position dependent. This means that immature root hair cells in the H cell position over the intracellular space become root hair cells, while cells in the N position become regular epidermis cells. You can see here that CPC is a positive regulator of root hair formation, while BHLH is a negative regulator of root hair formation. TTG can either promote or inhibit root hair formation depending on what it's interacting with. GL2 encodes a transcription factor that is a negative regulator of root hair formation. It only works in some cells due to its dependence on cell position. WER is a negative regulator of root hair formation and may also be a promoter of GL2. The WER mutation not only allows root hair formation, but also disrupts position-dependent pattern of GL2, leading to random expression of root hair formation. Question 2. What is the biological significance of root hair formation? You may be wondering why this all matters, little group. Root hairs are technically not needed for the survival of the plant, so why all the hype? Well, I can tell you that the development of root hairs is actually very important to the scientific community. Because root hairs are not required for survival, it is actually very easy to discover all the mutant phenotypes of root hair formation. If none of them are deleterious, you can actually find and analyze every genotype for the root hair phenotype. Due to this, we have been able to discover a lot about root hair formation and development. For example, we know that root hair development has high plasticity. Cells can actually change their differentiation programming in response to environmental stimuli. This can occur even if that cell has already begun to differentiate into a different cell type. We also discovered root hair morphogenesis is induced due to the auxin and ethylene presence. This plasticity to a variety of environmental conditions is unique, and it's interesting to learn the mechanisms behind. Question 3. What is the developmental process of forming root hairs at the mechanistic level? The developmental process is controlled at the molecular and cellular level by 3 main factors: genetics, hormones, and the microtubule cytoskeleton. The developmental process of root hair formation can be separated into 3 main stages: cell fate specification, root hair initiation, and tip growth. Cell fate specification is the first step. Certain genes, such as AXR2, SLR1, and CPC, were found to be responsible for promoting trichoblast cell fate, while others, like ERH1, WER, and GL2, were found to promote atrichoblast cell fate. In terms of hormones, ethylene was found to be a positive regulator of trichoblast cell fate. Mutations that altered auxin responses showed that the normal auxin pathway promotes root hair formation. Disruption of the microtubule cytoskeleton dynamics in cells before specification occurs can also affect root hair development. During root hair initiation, the growth habit of an epidermal cell changes from elongation to highly localized asymmetrical expansion on one side of the cell. Auxin and ethylene are important in establishing patterning of root epidermis as well as defining the exact location of the root hair initiation site. The earliest morphological indication of root hair initiation is the bulging of the cell wall at the root hair initiation site. As soon as this occurs, the pH of the cell wall drops, resulting in highly localized acidification. During tip growth, deposition of new plasma membrane and cell wall material is confined to the expanding tip. Elongation of these cells is associated with a tip focus gradient in cytosolic calcium. It plays a large role in driving and directing growth away from the surface of the root. Small G proteins appear to be regulators of tip growth as well. Interestingly, root hair tip growth is not disrupted by microtubule cytoskeleton disruptions.